The overall goal of this procedure is to knock down genes of interest in a growing population of E. coli.
Gene knockdowns are often used to explore basic questions of gene function. These methods may also have application in synthetic biology, for example, in silencing unwanted genes like virulence factors.
The main advantage of this technique is that it can be performed in batch cultures of E. coli without prior genetic modification, with results observable just after a few hours.
To begin, using a plasmid containing an sRNA expression cassette designed according to the text protocol, carry out site-directed mutagenesis on the sequence by first identifying the 24 base pair guide sequence in the expression cassette. Design and synthesize forward and reverse primers with short regions of homology to the existing sRNA cassette, flanking the new 24 base pair guide sequence. Prepare a 5 milliliter culture of E. coli in LB and antibiotics carrying the template sRNA expression phagemid. Grow the cells overnight at 37 degrees Celsius with shaking. After extracting and purifying the template sRNA phagemid from the culture, prepare 2 PCR reactions using 10 to 50 times the recommended amount of DNA template for the sRNA phagemid and high-fidelity polymerase. Prepare 1 reaction with the forward and 1 with the reverse primer. After using standard PCR conditions to run the reactions, combine the 2 reactions in a microcentrifuge tube. Then anneal the products by heating to 98 degrees Celsius in a boiling water bath.